Hey everybody, Miss Jatsy here again. I want to take a minute to go over some of the homework from last week because on Tuesday you're going to be having a quiz. So this picture is quite complicated, and it's always one of the things in biology that has so many pieces to it, makes it a little bit difficult to understand, and there's a lot of new vocabulary. So I thought we can go over this. You should have the work kind of passed back to you through Google Classroom if you need to make any corrections. But okay, so what you really see here, of course, is the DNA double helix, but it appears to be splitting into two. So this is an image of what's going on during DNA replication. And just to remind you, DNA replication happens during interphase of the cell cycle, and particularly it's the S part of interphase. It's the S stands for synthesis, which means to copy. Okay, so overall what we see is our double helix, which remember is running anti-parallel. So one side is the 5' side and the other side is the 3' side, and those primes refer to which carbon is the outside carbon on the molecule. The two strands are anti-parallel, meaning they actually run in opposite directions, even though they are complementary to each other. Okay, so I want to mention a little bit about the different enzymes and what they do. So first up here is helicase, and helicase's job is pretty straightforward. It's appropriately named. Remember that things that end in ASE are going to be enzymes. And enzymes are proteins, but they are particular proteins whose job it is to catalyze or to speed up a particular chemical reaction. So what helicase does is it unwinds, or a fancy way to say that would be to unzip. It unzips the DNA double helix. And the way it does that is by breaking the hydrogen bonds that hold the two sides together. So for example, A is going to be bonded with T and G is going to be bonded with C, but helicase's job is to break those bonds. They weaken the bonds to the point where they break, and then that allows the DNA to kind of start to unwind. So the helicase kind of runs all along throughout, making that happen. Okay, so once you start to separate the two strands, then you have these little purple guys here, and these guys are called SSB proteins. They're single-stranded binding proteins, and what they do is they kind of coat the DNA, so it's they're found all along. They've only drawn a few of them on the screen here, but they're all along the two sides of the now open double helix. And what they do is they prevent the bases from rebinding. So they're stabilizers. So they prevent the bases from rebinding to each other. What was the point of separating if the attraction between these guys is just going to bring them back together? So the SSB proteins will help prevent that from happening. Okay, next up is the big one, and that's called DNA polymerase. Poly meaning many. It's also an enzyme, so you can see the ASE ending there. Its job is relatively simple. What it does is it adds nucleotides. So nucleotides, remember, are a sugar connected to a phosphate group connected to one of the 4 bases. So a sugar, a phosphate, and a base. So it adds them. So if you have an A who no longer has a partner, let's say it's this one, right here, it's going to find a complementary base pair. So it's going to put an A back with a T. Now these nucleotides are floating in the DNA at all times, and you're constantly synthesizing nucleotides. So they're there, they just need to be assembled. So the DNA polymerase's job is to help kind of assemble them and put them back together. Okay, so how does that all work? There are some helpers. The first one's called DNA primase, yet another enzyme, so yet another type of protein. And its job is it's a sequence of DNA that makes something called the primer. So it makes a short RNA sequence, and I know we haven't talked about RNA yet, but we will in just a bit, sequence called a primer. Okay. So a primer, like if you've ever painted anything, one notes that you have to put a primer coat on first. It gets everything ready. And the case here is that the primer's job is to serve as the starting point for the DNA polymerase. So the starting point for DNA polymerase. In other words, how does DNA polymerase know where it needs to go? It's searching for the primer. Okay, so once it finds the primer, then it locks on and starts to synthesize in particular directions. Okay, so that's that one. I realized I had polymerase on there twice, so just ignore that for now. I'll skip down to ligase. Ligase's job is it's like the glue, so it seals the strands back together. And it does that by adding some of those phosphate groups back. So they add some of those stabilizing molecules. Okay. One other one, and this is my favorite enzyme, if I could choose a favorite, is called topoisomerase, and it's this really big one here at the top. And I wish I was there to show you in person how it works, but I did link a video to it in your notes, so watch the little video because it would have been the demonstration that we would have done together in class. But basically, you know, if you're separating the strands of DNA and DNA is twisted and you start to pull the two strands apart, basically you're gonna pull them apart and then they're gonna get stuck. You're gonna have so much tension in the line that it can no longer unwind anymore. So topoisomerase's job is to correct the over and underwinding of DNA. Another way to say that is that it cuts the DNA double helix. It literally just cuts it and then it releases the tension. It has to exist because if you just continuously pulled it apart in many different directions, because remember there are many origin sites where this is happening, eventually you're not going to be able to unwind it anymore because the twisting has gotten so tight from the end that you are not pulling on that everything would just kind of freeze up and get stuck. So topoisomerase is kind of cool how that one works. So please, please, please try to watch that video. It's only like a minute long. There's a link to it in your notes. Okay, now the tricky and the last part here is to identify what's going on in these particular sequences. Okay, so one of these strands basically is going to synthesize everything faster than the other strand, and we call that the leading strand. So the leading strand, remember, is doing everything faster, and another way to say that is it's synthesizing it continuously in one direction. The lagging strand is going to be a slower synthesis, and the reason it's slower is because it's fragmented. Because DNA polymerase only moves in one particular direction, and the two sides of the double helix are not moving in the same direction, they're anti-parallel, only one side is going to get the action fast enough. So if you were looking at a picture and trying to identify which one is the leading and the lagging, you have to look for one thing in particular, and they're called Okazaki fragments. Okay, so fragments are like little fragments of made or synthesized DNA. So when you see the fragments, this is always going to be the lagging side, and the other side will be the leading strand. Okay, another way to think about that is the direction that things are going. Your leading strand is always synthesizing toward the fork, where it splits, the split in the road. So it kind of just, this DNA polymerase will just continue to move along as this continues to open and open and open. So it's just a continuous movement. Whereas on the other side, synthesis is going to take place in this direction, but then it also has to kind of backtrack and then redo and fill in the pieces it misses. So you get all these like fragmented pieces at the end that eventually all get filled in, but it's just a trickier situation because polymerase only adds bases in a particular direction. So if ever on a test or a quiz, you're asked to figure out which strand is doing what, note that the synthesis is happening in two very opposite directions. Okay, I think that's good for now. I'll see you in a bit.